Hi, I'm Leslie Hansen. I am an MD MBA student at UVA. My project this summer is also with Sonodynamic therapy, looking more at the FDA approval process to get clinical trials and what is currently going on along with landscaping.
So before delving into sonodynamic therapy, I wanted to talk about photodynamic therapy since it's a more established procedure and SDT is derived from it. So PDT involves the use of light and a non-toxic photosensitizer to destroy cancerous cells. When the light and the non-toxic photosensitizer are combined, they produce cytotoxic effects to kill those cancerous cells. PDT has been used for the following indications: pancreatic cancer, bile duct cancer, esophageal cancer, lung cancer, and certain skin diseases. Some limitations associated with PDT are that since light cannot travel very far to tissues in the body, PDT can only treat superficial tumors that are on or near the surface of the skin, specifically 5 to 7 millimeters from the surface of the skin. So advantages and limitations. So compared to normal current cancer therapy, such as surgery, radiotherapy, and chemotherapy, PDT results in minimal damage to surrounding healthy tissue, meaning that it's a much more focused treatment. Additionally, there's faster recovery from the procedure. It can often be done as an outpatient procedure, and there's little to no scarring in the treated area. Unlike radiotherapy, it can be repeated several times at the same site, and it is also cheaper compared to other treatments. Again, one limitation I mentioned earlier is that it has a shallow penetration depth, and some patients can also develop light sensitivity after a PDT procedure. So here I wanted to talk about sonodynamic therapy. So SDT is an emerging approach that is non-invasive and offers the possibility of eliminating tumors. So in SDT, we use photosensitizer and ultrasound instead of a photosensitizer and light as used in PDT. So the method involves the interaction between ultrasound and non-toxic sonosensitizer, and when these two are combined, this exerts cytotoxic effects at the tumor site. And some relevant mechanisms that I will go over in this presentation are the cavitation effect, sonoporation, and Roche generation, which is reactive oxygen species. So advantages of SDT over PDT. One way that SDT covers a major limitation of PDT is that it is able to treat deeper-seated tumors. So ultrasound can be focused within those tissues at a discrete point. Additionally, SDT allows for selective sensitizer uptake, and that means that the SDT allows for SDT targets cancer cells instead of just normal cells, making it a much more focused treatment. So the cavitation effect. So cavitation is the creation or expansion of microbubbles in a liquid or gaseous medium. So as acoustic pressure is applied, microbubbles start to oscillate. So higher acoustic pressures result in violent shock waves because these microbubbles start to collapse and expand very rapidly, and this can cause mechanical damage to surrounding tissue. And this is a phenomenon called inertial cavitation. We also have stable cavitation, which is smaller oscillations at lower pressures. Stable cavitation also has a damaging effect, so to speak, because small oscillations result in microstreaming, which means there's a movement of fluid around those bubbles, which also creates pressure in the surrounding area. Another mechanism of action is sonoporation, which is observed in both inertial and stable cavitation. When cavitation basically results in the creation of micropores in cell membranes, and these are little holes that allow for increased cell membrane permeability and allows for the efficient entry of chemotherapeutic drugs to target tumor cells. So this picture, I think, beautifully represents this concept, and this is actually from the Focus Ultrasound Foundation website. So we have cells that are surrounded by drug molecules here. Focus ultrasound causes cavitation, which results in little pores in these, cells, and then the drug is the drug is able to go into these cancer cells, and then these cell membranes close, and then the drug remains in the cell. So now we have reactive oxygen species generation. This is really important because the sequence and combination of sonochemical reactions serve to produce the desired effect of tumor ablation. So there's three areas where these sonochemical reactions can occur. One is reactions at the interior of collapsing bubbles, which result in pyrolysis, and this is something I'll go over. There are reactions at the bubble liquid interface where there are strong pressure and temperature gradients that are created, which can serve to destroy and decompose surrounding solutes. And there's Ross generation from bubble collapse induced by inertial cavitation as well. So reactive oxygen species are generated by a combination of low intensity ultrasound, molecular oxygen, and a sonosensitizer. So increasing temperatures and pressures during microbubble implosion from inertial cavitation release heat and light energy. And what this does is that it concentrates energy to the tumor site. There are two concepts related to this where they're called sonoluminescence and pyrolysis. And sonoluminescence is the emission of light from imploding bubbles activated by ultrasound. And pyrolysis is the decomposition of sonosensitizers, which occurs due to high temperatures from inertial cavitation. And this last concept basically results in the creation of free radicals and other substrates that result in the creation of Ross generation. And so Ross generation results in many cytotoxic effects, including high oxidative stress, DNA damage, apoptosis, and lipid peroxidation. So Ross lipid interactions can basically disrupt the normal lipid bilayer structure of the cell membrane, which of course results in cell damage. And SDT also promotes apoptosis by the upregulation of genes BAX gene and downregulation of BCL2 gene. And this is really important because BAX is responsible for releasing cytochrome C from the mitochondria, and this is an apoptosis promoter, while BCL2 is responsible for the opposite effect, where it inhibits the release of cytochrome C from the mitochondria. So it makes sense that we want upregulation of BAX. So more on SDT. So SDT mechanisms induce anti-tumor activity. And one example of how this is done is the transformation of M2 to M1 macrophage. And this is important because M2 macrophages are anti-inflammatory and promote tissue repair, while M1 macrophages are pro-inflammatory. They release pro-inflammatory cytokines and result in anti-tumor activity. So it's good that we have M1 macrophage transformation to destroy those tumors. And SCT also controls angiogenesis, and this study with Gao showed that SCT prevents neovascularization. And this is also important because we don't want the creation or formation of blood vessels to support those tumor cells. And this study also showed an inhibition of endothelial cell proliferation and migration. So this figure, this is kind of like a summary figure because this kind of showed the SCT mechanism. So the first one you can see is pyrolysis. The second one is the creation of light through sonoluminescence, which results in the creation of Ross and then cancer and cell death. And lastly, this third pathway is basically compound affinity for the plasma membrane, which basically results in ultrasound-induced mechanical stress, and that also results in cancer cell death. So all three pathways result in cell death.
So SDT is a combination of two things, as we said. It's the ultrasound and a sonosensitizer. And so there have been two main sonosensitizers that have worked. One is fluorescein. It's an organic compound. It accumulates in the areas where the blood-brain barrier is impaired in the brain. So it's been used a lot for blood-brain barrier opening. It goes through the body very quickly. It is does not concentrate in healthy tissue. And the ultrasound activation of fluorescein causes damage through ROS, so through free radicals and the singlet oxygen. Particularly the study by Francisco Prada showed that there's noteworthy uptake in cerebral tissue and there's no significant issues elsewhere in the body, so it's very safe. The other one is this 5-ALA, and it is the most common agent used for SDT. It is a porphyrin-based. It metabolizes into this active PPIX. It's extremely selective. This is why it's interesting, because it really only concentrates in tumor cells. Many of the preclinical studies have shown that it has decreased tumor cell viability. It induces apoptosis. It's also very safe, and it's been used in previous brain cancer studies as a indicator of where the tumor is. So this is all well and good, but the question is, what do we do with all the science, right? So SDT is currently being studied for a couple different indications. In particular, we have GBM, melanoma, metastatic melanoma in particular, prostate cancer, and pancreatic cancer. And all these are ones that are deeper tissue. So that's the benefit of that. It can impair tissue way deeper in the brain through larger amounts. So again, what do we do moving forward? Well, the benefit of this is clinical trials. We've focused primarily on GBM. So for those who are not familiar, I don't know how aware you are of GBM, but it is a very devastating tumor in the brain. It often recurs. And so the way we could look at this trial, if we're looking at sonodynamic therapy, would be two phases. We can look at the change in the ultrasound parameters, or we can change the drug. We were looking at all the different sensitizers. What we really want is one that targets purely the abnormal tissue. We don't want to impact the surrounding tissue, right? Because you're tunneling down and there's healthy tissue above the tumor. So really 5-ALA is the one that works for this, because it's the one that concentrates in the tumor. Our plan at FUS would be a joint trial with NapaFUS. They're a Taiwanese firm. We have a strong partnership with them. Their particular ultrasound is a multi-channel hemispherical phased array. So theirs is circular scanning. The idea is the same as concentrated ultrasound. So in order to get this off and running, we have to get FDA approval for any clinical trial. So the problem here is that we have both a new device and a drug that are be studying for new uses put together. So this is considered a combination product under the FDA that's governed by this Office of Combined Products. And while it is a combined product, it only goes through one pathway. So one organization takes the lead. So generally, you only need to file either an IND, so an investigational new drug, or an IDE for a new device. It's based off of, their designation is based off the primary mode of action. So whichever, either the device or the drug, impacts the treatment the most. So SDT and photodynamic therapy, in particular, photodynamic therapy historically has been through the drug pathway. They say the primary mode of action is through the drug, the light activates the drug, and so it's the drug. Sonodynamic therapy recently has also gone through the drug pathway. So there's a trial currently at Faro. It's a first-in-human study. They're looking at sonodynamic therapy with 5-ALA. They were only looking at recurrent GBM. It's going to be up to 30 patients. They're given intravenous ALA 6 to 7 hours before SDT. So these patients are already scheduled for resection. And you can see in the photo, this is what 5-ALA was used for already. It highlights the tumor, it concentrates the tumor. So it's already approved for that, but it's not approved in this indication. And they're looking at two different ways. They're looking at dose escalation and time escalation. And really they're measuring biological tissue changes to see whether or not this is effective. So we know that this trial can get off the ground. They were approved through the drug pathway, partly because they're using InsightTech device that has already been approved by the US FDA for blood-brain barrier opening. So special considerations for us in this case is that the Navifus device has not been previously approved in the United States. The counter to that would be that the FDA has approved previous these devices. The problem, though, is that the technology varies so widely between companies. So while it is the same method of action, they're very different. So we can't guarantee that the FDA is going to come back and be like, yeah, it's fine, check. The other thing is that most of Navifus clinical trial that they have is all from Taiwan. And so it's unclear how much supporting data from Taiwan and international is going to weigh for the FDA as opposed to having a blood-brain barrier opening study in the US, like Incytec did. The other thing is that, is that we're going to be an add-on site for when this trial is up and running. This is going to start in Taiwan through Navifus, so we're waiting on that. But that also means that they will already have had Taiwan FDA approval. And so the question is, will there be any consideration in the US that there's already been an approval by an outside FDA? And the third very important special consideration is that the US FDA requires the application to be sponsored by a US person, which isn't an issue for us because we'll be partnering with Dr. Sheehan at UVA, and then we also have the FUS Foundation. So there are people that could be the primary US sponsor on the application, and we can import the device. But really the next steps of how do we move forward with this? How do we get this trial up and running? We're still waiting Taiwanese FDA approval. We're still waiting on Navifus. That's kind of on them. Once we get approval, once they get approval, then we can really move forward. In the meantime, we can also have an informal conversation with this Office of Combined Products, and then we can submit this thing called an RFD or pre-RFD. And basically what that is with the Office of Combined Products, you submit an application and they will tell you, go drug or go device. And they have two different ways to do it. There's a pre-RFD, which is non-binding. It's a little more informal. And then they have the RFD, which is binding. So if they give you a decision under RFD, all future communication is done through that pathway. The difference is, depending on whether or not sponsor wants it completely binding, if something changes, you're just jumping through paperwork and hierarchy and all that. But they will give you a decision supposedly within 60 days. And this is sort of the distinction between RFD and pre-RFD. Really, RFD is the very formal one. They came up with pre-RFD relatively recently because people wanted a more informal decision. And what you have to do is you have to describe your product. You have to include how it's made. You have to have all the supporting data, both preclinical and clinical. You have to explain the mechanism of action and how it's going to work. You would have to describe related products. And you have to do all this for an RFD in 15 pages. So the pre-RFD is kind of like, testing the waters a little bit. So you can include this, or you don't have to. But so that's kind of what we're doing at the moment.
Yeah, and we hope, for our next steps, we hope to get involved in Dr. Sheehan's lab for metastatic melanoma. And one more slide. This is a Lauren appreciation slide. So she's amazing and literally supported us throughout this entire time. I'm sorry if you don't like this picture.
She's a super hard worker. She's absolutely undeniably gorgeous.
She's fit. I don't know how she's that fit, but I don't know. She's a superwoman.
She's really practical. Really nice.
I love you guys. Good job.
Could have done Palovich too.
Oh God, that would be too many letters. Are there any questions before we sneak off this stage?
People on Zoom, just unmute
Yeah,
yourself.
does anyone have anything on Zoom either?